Recording in progress.
طيب السلام عليكم ورحمة الله. أسعد الله أوقاتكم يا رب بكل خير. Welcome all and thank you very much for being with us today. Is there any question or any concern? في أحد عنده سؤال أو استفسار؟ طيب الحمد لله الذي علم بالقلم علم الإنسان ما لم يعلم والصلاة والسلام على خير معلم الناس خير محمد. And welcome all in our meeting today while we are going to discuss the microbiology aspects in wound care. As someone interested to work in the field of wound care, what are the minimum required informations and the aspects that I should have while I'm dealing with the wounds and the conditions related to it. In general, while we are talking in microbiology, we are describing three aspects that can affect the wound care. There are bacteriology, or the bacteria related to the wounds, and there are wounds related to fungus, and there are wounds related to viruses. In today's lecture, we will concern about bacteriology, and later, inshallah, we will go for the other microbiological aspects that affect the wound care. Let's start by the bacteria, or the bacteria related to the wounds. We already understand the negative impact of presence of bacteria in wounds, and this can implement or lead the wounds to get healed badly progress. In general, the physical barriers like the skin and the mucous membrane help to keep pathogens out. So the bacteria already unable to penetrate this physical barrier, subhanAllah, that the creator gave to us. But on the same way, when this barrier, such what happened during the wounds, get injured, so the bacteria get the access. And the variety of specialized cells, when these variations or this injury happen, already designed to stop this bacteria from playing around and destroy the body. That's why we have the immunity system, and the body works against these invasions of microorganisms such as the bacteria. But our understanding for what's going on while we are dealing with the bacteriology help us to support the body during this fight and during this invasion. So let's here go and start to define the bacteriology as a science and its impact on us in the wound care. When we are talking about the bacteria, we are describing as a unicellular microorganisms. Unicellular, that means we are talking about individualized cell. The cell able to live within the community, but each cell has its own independence. Bacteria live in a vast number of places, and they are categorized. And this categorization can help us while we are dealing with the bacteria in the wounds, how we will work against it, or at least support the body in this fight and in this condition of invasion. History of bacteriology, I'm talking here about the bacteriology as a science, not the history of bacteria itself, because the bacteria, subhanAllah, there are a lot of evidences shows that the bacteria in the planet even before presence of humanity. فالبكتيريا بدأت قبل البشرية نفسها. لكن the bacteriology or developing of this science started by Anton van Leeuwenhoek, which started by discovery of the microscope or creating the microscope in 1600s. And this is the first microscope that used at that time, and it was the shock. They discovered living cells, small, cannot be seen in the naked eye, and need this microscope. And you can see here the first draw for what they say by using the initial microscope. They saw different type of bacteria. They start to draw it to show for us what did they see. The big jump happened by the development of the germ theory, and it was by Louis Pasteur who developed or started to initiate this theory, and he discovered the pasteurization أو البسترة هنسميها بالعربي in the 19th century. Then this is the portrait for the Louis Pasteur, how he was working with his lab for isolating and defining the bacteria. Robert Koch established also additional milestone in the bacteriology aspects, and he started to define or find the connection between presence of bacteria and some signs and symptoms appears for the patient, which is the disease, such as tuberculosis and cholera. This is photo for Robert and start to differentiate and name the bacteria. They rely on the names that come from Greek and Latin origin, which make the names in the bacteriology, the people who are already getting specialized in this field, getting or sense of hardness to study this course or this specialized topic. Either you come from Arabic background or from the modern English or whatever your origin language or native language. The problem that comes in the bacteriology when we are students and study this topic, that's from the names, because it's a lot of technical names and they used already dead language, such as Greek or Old Greek and Latin. That's make the names a little bit difficult. We will try in our scope in the wound care to work to differentiate it and make it easy for us to remember, at least for the topics that will be frequently appear in the world of wound care. This is the normal cell, how it looks like. The human cells, you can see the complexity of molecules inside. We see the main characteristic of the developed human or animal cells, that the nucleus is already isolated. It's not within the context of other parts of the cell. While when we come to the bacteria, we're talking about much more simple form. It's directly just a genetic material, is swimming in cytoplasm, and it's covered by cell wall. So we can simply say that the bacteria, it's lived cells directly with the genetic material that swim within the cytoplasm, and there are cell wall to protect. So this is the simplicity of the bacteria. And this simplicity make it a positive part regarding to its ability to penetrate and make the diseases, and challenging part, because of its symbol, it make it easy for this bacteria to swim and get the injury within the targeted object. In the science of bacteriology, you will hear the term prokaryotes, which divide the bacteria to bacteria and archaea. Now, in this lecture, we will use the term of bacteria because archaea is not a concern for diseases and producing the problems that related to us in wound care. Right. Let's start here to define the bacteria. What are the characteristics of the bacteria? What are the characteristics that important for us here to know? So simply, we talk about cell wall. So, and this cell wall simply will provide the shape and the protection. And the shape of the bacteria is very important for us because it will give important part related to how we will define and differentiate these kinds of bacteria, because a lot of types of bacteria already come within the target of the shape. So the shape of bacteria plays an important role for us while we are dealing with the bacteria, as we will see now. And there are the plasma membrane, which is the control of the entry and the exit of the substance. And it's important for a lot of items related to the antimicrobial aspect. And there are the cytoplasm, which contains the genetic material and cellular mechanisms. There is also the ribosome or ribosomes, which is the site for protein synthesis. And there are the flagella, which the movement part in some bacteria. And as we said, the shapes which come from the cell wall can be varied between cocky, bacillus, spiral, and so on. Here, these are the major shapes that you will see the bacteria on it. So you can see the shapes that depends, like for example, the Staphylococcus pneumonia, Clostridium, etc. These are the shapes important for us or repeated for us in the field of the wound care. It depends on the type of bacteria. So you will see here the bacilli, the cocci, and the spirilla. And this classification depends on the shape of the bacteria. So the most frequent or the most repeated type is cocci, عنقودي بالعربي. These bacteria are spiral or round. An example of it, Staphylococcus aureus or Staphylococcus epidermis, which look small clusters of grits, يعني زي عنقود العنب. Bacilli, these are root-shaped bacteria, like little cylinders, and examples of Escherichia coli. And these examples in the field of the wound care. We will see also spirilla. The bacteria have a spiral or corkscrew shape. And examples, the Hypobacterial pylori, which is associated with the stomach ulcer. Its problem also can be associated with a stoma or peristomal infection. Vibro, these bacteria are common shaped or curved root, and examples of Vibro cholera, while Listeriococcus, which is long, thin, flexible, spiral-shaped bacteria, مثال عليها Streptococcus pyogenes, which cause syphilis. Here we will note the flagella. This is part of the bacteria that not appear in all type of bacteria, some type of bacteria. But what are the differences happen when the bacteria have the flagella on it? Simply, it's responsible or support the bacteria in movement. And this is important here to notice the impact of the ability of the wounds for the movement, and this movement can lead to change the location of the bacteria, and this location of the bacteria, that means the wounds can move or swim around. The flagella had variance in its shape, so it's not only one foot or one source of movement. It can be varied, it can be all over the cell wall, it can be in one point, and at the same time, it can direct the bacteria toward the food and resources. The most or the bacteria with the highest flagella, known as Tadopli, and there a lot of tails can cover the cell wall, which means that the bacteria is very fast in movement. The bacteria varied in its size, so we are talking about 0.1 micrometer until 5 micrometer. And these bacteria in general, they have the British to movements. Here we can see the impact of the ability of the bacteria in movement. So this bacteria, because of its ability to move with the flagella, so it's distributed. And this bacteria, a kind of the bacteria that's hardly to be moved or jump around, so they will be colonized or stay in each other. The other characteristic of the bacteria is the ability to division or growing. Let's see for this example, Clostridium perfringens. This is the fastest growing microorganism ever known. This bacteria, under optimal condition, able to reproduce itself in 6 minutes and 20 seconds. And if you have 20 piece of cells of this bacteria, it can be doubled within 6 minutes and 20 seconds. And this is what they call it the astronomic rates or سرعة صاروخية in the development. This means they can produce a huge amount of itself in piece of seconds. So in the other or in the following 6 minutes and 20 seconds, you are talking about 400 pieces of bacteria, then this will be end by a huge amount of cells in piece of hours. Luckily, يعني Clostridium perfringens is not the same or it doesn't appear frequently in the wounds. But unfortunately, we have other types of the fast-growing bacteria that can appear frequently in the wounds, which is the Escherichia coli and Staphylococcus aureus. And Staphylococcus aureus, it's one of the headache. We will discuss about it at the end of this class, that it makes a challenging time for the people working in infected wounds. The generation time of these two can be around 20 minutes, so they are moving fast. Yeah, wallahi, you are dealing with the sacral wounds, and there are infection come, Escherichia coli come from in the sacral wounds from outside, maybe from the GI tract, from any other resources. Then when you clean it up, but unfortunately there are a small piece of Escherichia coli stay here. Then after 20 minutes, this small will be doubled. And when you come back after 1 day or 24 hours, you are talking about almost the same amount of Escherichia coli had been reproduced again until you have your own strategy to work to state this number or make it down. The ability of bacteria to survive, there are a lot of evidences that talk about the ability of bacteria to survive under a very challenging environmental condition, under the volcano, under the water. In Jordan, we have the Dead Sea. There are the water with the full of salts that make the life to be impossible for the fish or for any other sea life. But for the bacteria, it's suitable. There are a lot of type of bacteria living there. How the bacteria already produced? We're talking about the binary fission, which means the normal reproduction of the bacteria. When we have two bacteria, then the division will happen, and we will end up by two pieces of bacteria, and these two also will be divided to two, and so on. So this is how the bacteria divides. The same way that the bacteria vary in its shape, it's vary in the genetic variations, which means that the same for the genetic materials, they have a variation inside this genetic material and its ability. One of these variations, it's what they call it the conjunction. There are a kind of bacteria able to do the conjunction, and this is a kind of, or give the ability of the bacteria to contact with the other bacteria and transfer kind of genetic material between these two bacteria, and that means there are a kind of learning, maybe we can call it, which lead at the end for the what known in the resistance, or one theory or one strategy that the bacteria can use for resistance. There are also transformation when the bacteria can attract material from outside and insert it within the genetic material, and this can be for transformation, and this also can lead to the resistance, or they can lead for other suitable or useful production in the pharmacology for some kinds of hormones that use transformation mechanisms. And there are transduction when the transfer of genetic material affected by the viruses, just as the bacteriophages, when there are a transfer happen for the genetic material associated with the virus insertion or presence of the virus, which end to destroy the bacteria by kind of viruses in this way. Still, there are challenging ways to classify it, how we will deal with this. So one of the big achievements happened in the world of bacteriology, which is the gram stain. We hear a lot about gram positive, gram negative. So what is gram? Gram, it's a type of stain. It consists of kind of staining this bacteria, and they discovered a unique result. This kind of stain classify the bacteria to two colors, either purple or pink. And this gets interest for the scientists to classify it, why the purple happened, why the pink happened. And then the two terms already associated with it, which is gram-positive and gram-negative. Gram-positive, they associated with the thick cell wall, while gram-negative come with a thin cell wall, and the result after this stain to be pink for the gram-negative and purple for gram-positive. This is how it looks like, and this already developed by Danish medical doctor. His name Hans Christian Gram, and from his name, this gram stain start to be known. So this is the name of the person who created this stain in 1884. This is the shape how it looks like. So gram-positive, that means there are thick cell wall. So the aspects of the stain, when it's come, it's refused to get out. While in the gram-negative, the stain, when it's come, able to get out easily, but it may leave the stain of the cell wall in this way. So that's why it's gram-negative. The characteristic here, it's easy for us now to differentiate. Gram-positive, that means we're talking about thick cell wall. بينما in gram-negative, we are talking about thin cell wall. And this already getting in examples to differentiate it in this way. Wallahi, for example, we know the gram-positive now, it's a staphylococcus. So directly, when I hear the term staphylococcus, now I hear staphylococcus aureus or staphylococcus epidermis, it's a gram-positive. That means staphylococcus will be in a coccus, grape, living together in this way, and also has thick cell wall. While when I told you now the gram-negative, an example of it, Escherichia coli, so Escherichia coli will have thin cell wall compared to the Staphylococcus aureus. And this is part from how we will deal with this characteristic of the bacteria. For example, let's take the MRSA. This is the headache that anyone work in the field of healthcare, maybe in the wound care, will have these challenges, which is the methicillin-resistant Staphylococcus aureus. So the first information I will talk about MRSA, it's a type of Staphylococcus aureus. So we're talking about Staphylococcus عنقودي, come on the grape shape, thick cell wall, and it's methicillin resistant. That means they have developed their own way to resist the antibiotics. It's a gram-positive, and it belongs to Staphylococcus genus. So the gene inside Staphylococcus, which consists of the gram-positive bacteria. That means all Staphylococcus species, including MRSA, are gram-positive. And this is important here to notice for the people working in the wound care, that no Staphylococcus come as a gram-negative. Until now, it's not discovered. We assume that there is no any Staphylococcus aureus gram-negative. So all Staphylococcus aureus is a gram-positive. That means thick cell wall. Now, this is the way, because there are frequent questions come to us from the people working in the field of the hospitals and dealing with the patients with infection, that can bacteria have both gram-positive and gram-negative. يعني احنا هلا حكينا انه الستافيلوكوكس all is gram-positive. Is there a kind of bacteria that can have both, يعني type of the same name of the bacteria have these two options as gram-positive or gram-negative? Yes, but under strict conditions. We don't want to come on that in this side. But for us people who are working in the wound care, the most frequent type of bacteria that come to them اللي هي Staphylococcus aureus, Staphylococcus epidermis, in some cases Staphylococcus spirochetes, all gram-positive. So عشان then I will guide you now how we will deal معاها. So Staphylococcus كلهم gram-positive. Gram-positive, special characteristic for it as we will come. There are some bacteria can have these variations, and frequent or can be seen also for wound care. يعني bacteria can species of this bacteria can be in the condition of gram positive and kind of, but the same species, gram negative and presence in the wound care. Yes, mainly اللي هم زي الإشريشيا كولاي والسودوموناس أريجينوزا. هذول اللي can become within staphylococcus gram positive والgram negative. طيب بما انه احنا ركزنا now and we come here and take time. Okay, gram positive, okay, gram negative. طيب, so what is the difference now? Okay, we know that gram positive or gram negative. Wallahi, why we concentrate on this one? What are the impact? Come to me to know these differences. First, it's the thick in the cell wall, the thickness of the cell wall. Here, very thick, so the bacteria will come and the cell wall is thick here. So this cell wall is thick. طيب, الثيكنس هذا شو الامباكت تاعته on me? Simple, the thick cell wall, that means higher resistance. So when we come to say that this cell wall, we wanted to enter antibiotic or enter antimicrobial, it takes time because there are a lot of layers already in this wall until it's come inside. So there are strength or strong point for this bacteria. Compared to the gram negative, is cell wall thin. So theoretically speaking here, والله الability to penetrate أسرع, right? This is true if we just think that this gram negative will stay silent. What did they discover? The gram negative, majorities of this bacteria are ready to produce what they call it safe zone for this cell. What is this safe zone? The bacteria will produce enzymes, and these enzymes will be able here to make additional protection for the bacteria. Unfortunately, majority of these enzymes also can be toxic and lead also for war that will be applied on such bacterial infection. Another way to classify the bacteria depends on the oxygen requirements. The bacteria that use the oxygen or cannot survive without presence of oxygen, call it aerobic or require oxygen. While the bacteria doesn't require oxygen, call them anaerobic. And there are fluctuative anaerobes, which the bacteria doesn't have any problem. The presence of oxygen can survive, and in absence of oxygen also can survive. So this is a kind of differentiations between these two types of the bacteria. In many cases, the bacteria that need oxygen will produce bad smell. So put this in your mind. The bacteria depends on the oxygen for staying. It will prefer to stay in direct contact up on the skin because they need this oxygen and usually, but this is not necessarily, but in many cases, this is how it's already happening. Now, is the bacteria causing the problem in the wounds? Definitely yes. But how this already happened? By penetration. So when the bacteria able to penetrate the barriers of the skin, that means activation of white blood cells, and directly the bacteria, when it's come, there are several scenarios how it already works until this happens. Let's say here in the normal skin, let's say this is the normal skin, and there are normal flora. The majority of the bacteria will stay as a normal flora on the skin. Staphylococcus epidermis. Staphylococcus epidermis is very important normal flora, even when we come to talk about the moisturizing and the health of the skin. This is very important part to keep it and save it. So while we already using the sunblock or moisturizing cream on the skin, we will use something safe on the microbiotic genetic codes of us and mainly for the Staphylococcus epidermis. What they are doing while there is no infection, they are fighting any other bacteria. So any other bacteria, when it's come, the Staphylococcus epidermis will not give them a space for them to stay. So if the bacteria come, they will fight. The same way they will help the normal skin to finish with the keratinocytes, the dead cells, and deal with it and make it easy to be penetrated through the air. The problem happens when there is injury on the skin. Unfortunately, the Staphylococcus start to remove these tasks and start to dive in the wounds. Now, Staphylococcus epidermis cannot move. It's not easy for the Staphylococcus epidermis to move, but they start to make the divisions and reproduction toward these wounds. These mechanisms make the Staphylococcus epidermis such as the stairs, little daraj, for the other bacteria and fungus to come on. The bacteria, the other type of bacteria, it's not easy for them to come and dive in the wound solutions and the exudates here and the enzymes, so they will die. But the Staphylococcus epidermis can stay on these enzymes and connected to each other in a way that can be originated from the peri-wound area, which make it easy for Staphylococcus aureus, other fungus to come, and they, in this case, Staphylococcus epidermis, play the wrong role. They here start to help the other microorganisms to destroy the body and destroy the mechanisms of it. These conditions that come under the pathogenesis or the ways of how the bacteria produce the disease. It's a penetration, survival under this condition, start to recruit and eliminate of the other parts of mechanism. Now the body will not stop, so they will start in short-term infection. Then there are a lot of or activation of the inflammation. That's what happened, and this will happen here on the acute, or start in the acute infection, or start the response of the body to fight. What happens usually after in the wounds? We're talking about specifically about the wounds. So this is the wounds. The bacteria start to come and accumulate. Then the white blood cells start to penetrate and come. So what happened in the blood investigation? Here, if I go now to see the CBC of the patient, because now there is infection, white blood cells here come to the wounds. Oh, discover there are a lot of bacteria. Hello, call the center, now increase the production of white blood cells, and increase the production of absolute neutrophilic counts. Now in the CBC, absolute neutrophilic count increase the number of white blood cells and so on. This is all happened in the acute phase. But this bacteria start to develop their own strategies. We talk about the biofilm, talk about change from the white blood cells. That's why in the chronicity of the wound, this stop the process of increase the production of white blood cells will be reduced. This is important why in many cases that the patient come to the clinic of the wound care with infected wounds. Infected diabetic foot, infected. And this infection stays for time. So now it's in the chronicity side. But when you check for or bone infection, but when you check the CBC, the white blood cells within the normal limits. Why this happened? Because of chronicity and the behavior of bacteria that will escape from the white blood cells, which start to give signs for the body that in the chronic wounds, the white blood cells can know changes how it's already happened. But how the bacteria can do that? What are the factors in the wound itself can support the bacteria to play this role, to fraud the body? First one, any presence of any foreign material inside the body, either related to the wounds or whatever, it will produce this house for the bacteria to stay in. If you now ask anyone interested in nursing, how many days you keep IV peripheral line. They will tell you 72 days, 72 hours, which is 3 days. How many days you will keep the CVV line? They will tell you, for example, maybe 5 to 7 days. But based on what they come to say this 72 hours, what are the statistics or the evidence they take it for that we will keep the cannula for 72 hours? The evidence and the answer come from our understanding for the presence of this foreign body in the IV line as in plastic. It takes 72 hours until the Staphylococcus epidermis start to learn how they will build the biofilm on the cannula. That's why if we keep the cannula, for example, for more than 72 hours, there will be phlebitis, there will be cellulitis, and other problems. This is the same. Originally, it's come from the wound care. In 1960s, they start to concentrate more about debridement for the wounds. Here, we discuss about the debridement or removing the tissue management, and Allah, we should remove. We should remove necrotic. We should remove the stuff. But why we should remove? But let's keep it. If we come to the healthy patient, for you, and you get injury, and part from this skin get out in this way, what will happen in the normal situation? The body will do auto debridement. In this case, wallahi, if we have this necrotic, why I should remove it? Khalas, keep it until this to be removed by itself. The problem is not by presence or removing of the necrotic. This is what we learned from Staphylococcus epidermis, that this slough tissue or necrotic will be the base for the biofilm formation. So what happened in this case, the time or the change or the reason for making the debridement is get out this chance from the Staphylococcus epidermis and the other bacteria, of course, but mainly the Staphylococcus epidermis. So this is how the mechanisms can happen. This is the same what happened, for example, cystic fibrosis, problem with the patient's inability to breathe, associated with chronic osteomyelitis. And directly, when the bacteria develop the biofilm and learn how to do this biofilm, there is no antibiotic can defect it. And for the antimicrobial, there are a lot of questions mark about its ability only to defy or to destroy this biofilm. I will tell you a story, and I was in this story as a part within this scenario. Then this case getting influenced with a lot of investigations to understand what happened. There was a patient, female patient, 65 years old, and she come to the clinic of the diabetic foot with diabetic foot, but the wounds did not reflect severity that the patient complained of. الجرح ما كان بين انه really deteriorated. But the signs and symptoms that the patient have كانت near to be in the septic shock, fever, low blood pressure, tachycardia, typical septic shock. With the history, we understand that this patient underwent 5 years ago for knee replacement. طبعا directly it transferred from the wound care clinic to the emergency and admitted, was still followed by in wound care. At that time, they start to pin out that the knee that replaced already get infected. But how it will be infected after 5 years? And حتى with a lot of surgical site infection strategies with protocols, well, it take about 1 month. The previous guideline, they were talking about 1 year. But after 5 years, with C-reactive protein عالي كذا to discover انه هذا الwounds اللي كان هي مع diabetic foot, but then the problem start to be in the organ transplantation, the knee knee implant. Then, and directly the decision was they should remove in the previous knee. When they remove it and they match in pathogen results, they take it the pathogenic histopathology, and we take tissue culture from the wounds, we found a match. And the same infection that in the foot is the same infection happened in the knee implant. And the biofilm developed in the organ there. You know, the bacteria penetrate through the circulation until it reaches to the mechan there. وهذه اعطتنا انه this can be happen because of the nature of the Staphylococcus aureus biofilm implementation. It can be traveled by the blood to that place and so on. Anyway, the case, alhamdulillah, after they remove this knee, يعني getting better, but this can teach us how we will deal with the conditions related to the characteristic of the bacteria. To not get it in long details, we do the debridement because the necrotic tissue and the slough will be the base for the bacteria to develop the biofilm, and this biofilm can be deadly and lead for severe conditions in such cases. Okay? Until now, we talked in general about the bacteria. We did not come in details about what are the important or the most frequently kind of bacteria that can change our behavior while we are dealing with the wounds. Let's start by taking them by the first one, Staphylococcus epidermis. From its name, it's epidermis, that means associated with the presence in the epidermis. It's apart from the normal flora. And it's a Staphylococcus, and it's already gram-positive. So as we say now, gram-positive. So there are a thick cell wall. And now additional adding, it's co-glaze negative. That means it's not producing co-glaze. Now we will talk about it later. But there is no secretion for this enzyme, which may already coming in this shape. The stain purple in the grain stain and doesn't produce the enzyme the glucogenase or glucose. This is how it looks like. The Staphylococcus epidermis. This is the Staphylococcus. We'll come to that in this way. The movements, it's very hard for this bacteria to move or change its location. It takes long time until this happens. It's a gram-positive and it doesn't have the glucose negative. That means it's not produced the glucose. In this case, the movements is not there. What is this glucose? The glucose is associated more with the Staphylococcus aureus, which is a type of enzymes that can destroy collagen or to produce a fibrin clot. So it's very dangerous and very negatively impact when it's already produced on the bacteria. Staphylococcus aureus doesn't produce coagulase. This is when it's already positive. It will destroy and redistribute the fibrin gene in the body. What are the characteristics of Staphylococcus? Staphylococcus, it's fluctuated in the exogen, so it can be survived in both, and this gives it dangerous impact during the wounds. The Staphylococcus epidermis can stay on the skin with the presence of exogen. والله ما عندي مشكلة. If there is wounds that necrotic, so the exogen is not reaching for us, also doesn't have a problem and can survive. So it's fluctuated and can stay inside. As we say, it's apart from the normal flora, and it's already able to survive from the medical devices or the patient contacts. And you remember during the 5 moments of hand hygiene, the last moment when you touch the patient's surroundings, the Staphylococcus epidermis able to distribute around the patient's surroundings, device, cell bed, and so on. The majority of wound, it's harmless, and except for the immunocompromised patient. So it's rarely that Staphylococcus epidermis can injure the patient in the normal condition or the human normal condition if there is no wound. But if there is wounds, it can form in biofilm, and this biofilm can be lead for the difficulties and business. Now, this is characteristic of biofilm cannot be consist from one type of bacteria. Now we will explain. But the initial bacteria that start to produce biofilm بنسميها على اسم. So we name it based on that. When I say now biofilm related to Staphylococcus epidermis, this biofilm consists for more than one type of bacteria, but the Staphylococcus epidermis, they are the one initiate bacteria. They can produce sepsis and endocarditis. It can come from the wounds. The other types or second type of the bacteria are the Craniobaccterium species. And the Craniobaccterium species, it's gram-positive, non-motile, root-shaped bacteria that depart from normal flora. The second frequent type of the bacteria, the normal flora after Staphylococcus epidermis, and in the same way can be negatively impact on the chronic wounds. It's a bacilli, the cranium bacteria, cannot move, non-motile bacteria, cannot move by its own, and it can be aerobic or anaerobic, so it's fluctuative bacteria. This is how it's already made, the cranium bacterium, or species بتاعتها. There are 4 major types can happen in the chronic wounds. We have the Craniobaccterium sternum, the Craniobaccterium amylobacterium, the Craniobaccterium jejunum, and Craniobaccterium cirrhosis. With cirrhosis, it's apart from when we have a hyperproduction of the keratinocytes, القشرة أو الصدفية كثير من الأسماء اللي بنستخدموها cirrhosis. The amount of bacteria in this case will getting much more increased and make a change to allergy for the patient. The Craniobaccterium also can support production of the biofilm. It leads for prolonged inflammation. The problem in the Craniobaccterium, it's easy to penetrate, but they have deadly epithelialization and they are able to destroy newly formed tissue. So it will lead for higher rate of destruction in the new epithelialized tissue, and that's responsible for the gaps that can be seen in the granulations. Granulation tissue not come on the same level. Usually it's associated with the Craniobaccterium. So we talk about Staphylococcus epidermis, Craniobaccteria, and now the moisture. أول وحش, Staphylococcus aureus. The Staphylococcus aureus, it's come from the gold color, so that's why this is the shape. It's from outside. There are similarities with the Staphylococcus epidermis by the shape, because of that it's a staph or Staphylococcus, but it are more problematic for us. Gram-positive, it's a copy, but it's a localized positive. That means they are able to destroy collagen. And this is the dangerous in Staphylococcus aureus in the wounds, which can clot the blood plasma and even destroy collagen in the wounds. The pathogenicity of the Staphylococcus aureus is high. A major cause of the wound infection due to its ability to produce a variety of toxins and enzymes, and it can produce or support production of biofilm in higher rate. So directly can support production of the biofilm. Now there are different types of the Staphylococcus aureus. The worst, it's MRSA MPR, يعني methicillin resistant Staphylococcus multiple drug resistant. أو the worst اللي هي methicillin resistant Staphylococcus aureus ADR, or what known as all drug resistance. This is really problematic kind of the infection. Before 10 years, we were talking about only MRSA come from the hospitals, but unfortunately, in the last 10 years, there are a lot of evidences about developing of cases or presence of cases, MRSA community acquired, which is red signs. I think there are a lot of national strategies start to control using of antibiotics. We hope, inshallah, in Jordan, we are going to produce such a policy to prevent or prohibit any antibiotic distribution for the community without or adding more restrictions on the antibiotic to fight such kind of bacteria. What is the impact of MRSA in the wounds? Now, it's variant than the Cranibacterium Staphylococcus epidermis. And Staphylococcus epidermis, the Cranibacterium, almost all type of wounds will have it. The Staphylococcus aureus, no, not all the type of wounds they will have it because it will not easily to come as an infection on wounds. But when it's already appeared in the wound, it will lead for prolonged inflammation. والتيشو دامج higher rate of tissue damage لانه في عندي في الستافيلوكوكس اوريوس الفا توكسين وانزيمز can destroy it like this. وهذه كمان also the Staphylococcus aureus very tough on the immunity responses. So this is the third time. We talk about three types: Staphylococcus epidermis, we talk about Cranibacteria, we talk about Staphylococcus aureus, and number 4, Pseudomonas aeruginosa. Pseudomonas aeruginosa is gram-negative. This is the gram-negative bacteria. This is the first one we talk about. It's a gram-negative bacteria. It's a bacilli, and the problem in the Pseudomonas, it's flagella owners or motile, able to move. So this Pseudomonas aeruginosa, now all the previous types, within the wounds, it will stay in one area and found very difficult to travel within the wounds. That's why in the Staphylococcus epidermis infection, Cranibacteria, Staphylococcus aureus, it's easy to find in pizza shape that we know. In one kind of the wounds already getting infected, the other types already getting progressed because it's away from the bacterial concentration. Pseudomonas aeruginosa, لا. Pseudomonas aeruginosa able to move around in wounds. الكاركترستيك الرئيسيه بتاعت السودوموناس aeruginosa البيجمنتس والسميل. لانه السودوموناس هذه لانها جرام نيجاتيف، حكينا هلا الجرام نيجاتيف. So what they are doing to protect their own cells, they produce something to make this safe zone. The Pseudomonas aeruginosa produce greenish blue pigments اللي هي البايكاين and bivalve. هذه الكاركترز، هذا شكل السودوموناس aeruginosa, how it looks like بالديش بوتس. هذا how it looks like in the wounds. I'm sure you already see such kind of exudates that come within the wounds. في greenish color هيك بيكون in the wounds. Pseudomonas aeruginosa negatively impact the wounds better more than or in worse scenario, prolonged inflammation. It's because of this type of enzymes that the bacteria produce, it will lead for accelerated tissue necrotics and of course higher resistance in treatment. Escherichia coli. So who can still remember what other bacteria we discussed? Staphylococcus epidermis, Cranibacterium, Staphylococcus aureus, Pseudomonas aeruginosa, number 5, Escherichia coli, our E. coli. The E. coli, again, it's a gram negative, so we're talking about pink color, while it's already in Spain. It's a bacilli, typically appears in straight roots, usually أكثر واحدة واضحة في البacilli. The most obvious type of bacteria in the bacilli is Staphylococcus Escherichia coli, and it's already fluctuative, so it can survive with or without oxygen. Escherichia coli, it's increased the inflammation, tissue damage, and the problem in the Escherichia coli also is the ability to move, so it can be easily to penetrate from one location to another, and that's why biofilm can be seen more under sacral pressure insert because it's even can be generated or travel from the gastrointestinal system. But during this explanation, we talk about the term of biofilm. What is the biofilm and why the biofilm? It can here create more challenge. Now, we talk about the biofilm, but we did not explain in deep what is the biofilm. اللي بيصير عندي أنا في البايوفيلم إنه the same bacteria here start to work with each other, creating this mushroom head, we call it, and they classify their own cells that the bacteria will create this wall, and we call them here soldiers. And there are other bacteria will be like workers. These bacteria that come on the wall, they will start to not breathe, not do anything, rather than just only stand up and protect the others, which make it very hard on the other any antimicrobial or antibiotics to penetrate, and this gives a chance for the life inside. But the only important part here that they need non-living base قاعدة غير حية inside living environment. يعني we need something here on the base to be not alive, but all others around it to be alive environment. For example, مثلا we need dentures that the white stain that come on the teeth if you did not keep the teeth washed. So you will discover something like a white soft. This is biofilm. Or can be on the cannula, can be in the necrotic tissue, slough tissue, and so on. So this is the condition of it. How it's already developed, what are the development? Here we will see in this video the development of the biofilm. You can see this is bacteria living around, freely moving, but after time, there are something that will be changed on this bacteria, that they are initiating the biofilm model. So you can see here on this video how the biofilm formation, number of bacteria start to originate near each other, and they start to communicate and build this community. This community doesn't need to have one type of bacteria. It can be developed by more than one type of bacteria, and even some cases can be developed with fungus, يعني with cooperation with the fungus. Subhanallah, يعني we have آية في القرآن وما من دابة في الأرض ولا طائر يطير بجناحيه إلا أمم أمثالكم. They will start to create like in a city, that this city able to develop a kind of communication around it, and this communication will produce a kind of cooperation in the biofilm. This biofilm, it's a feature that can be seen in the nature, it can be seen on over the human. This is the condition that this cooperation of the bacteria that needed. If you go to the river or near the sea, and you will see some kind of elasticity of the exposed water or unmoved water around a river or around pool or whatever, this is biofilm. This taste or nature of the biofilm. بالعربي شيء يكون زحلق على حواف السودوموناس وحواف المية الراكدة. So this is how it's already developed. So at the end, what is the biofilm? It's from its name, it's bio, so it's something genetics. And this genetics start to be developed, this biological, and start to be developed in the layer. So when they come here to develop this mushroom head, in fact, it's not one layer, there are a lot of layers come inside, and this layer is already filled with the bacteria, but the characteristic of bacteria in each layer can be varied. There are a lot of development or a lot of discoveries and research, R&D working on this one, talking about one center now in Danish, work only on the microbiological condition of the biofilm in the wound care and other kinds of infections that can happen, such as cystic fibrosis and so on. As we see the plaque on the teeth, this white, dark substance that developed on the base of the teeth, this can be developed. It takes around 12 hours for 24 hours to be developed and can be produced in this way. It produced, and directly when this production happened, it's making a resistance for the white blood cells and the bacteria. This is the shape of Staphylococcus epidermis biofilm. So you can see this is the connection. They have this connection around it. And this is the Staphylococcus epidermis. This is the connection around it. And you can see now the variation in the type of the bacteria. There are kind of bacteria doesn't do anything. So the temperature of it, it's less. And there are the bacteria produce the energy. And these layers can stop the impact of any antimicrobial that I will put. Whatever here will happen, it will destroyed by a lot of layers. And this antimicrobial agents that I will put it here needs a lived cells, the same what we said in the silver. Silver requires the bacteria to breathe, open the gates so the silver will come. But if this bacteria did not breathe, closed all the gates, so how the bacteria or how the silver will produce? There are 3 stages for the development of the biofilm, which is attached, start to be developed, the bacteria know each other, start to talk to each other, then they develop this community and the growth, and subhanAllah, the detachment happens in the biofilm that they are split out. So when you come to see the microscopic way, the biofilm communities get out the new, when the number of cells increase, so they get it out in space. That's why the spread of the biofilm start to take it out. The most discoveries or the initial discoveries for the biofilm come in the 1956, so it's not all the discoveries. And what did they do in this study? They bring a number of 7.5 Staphylococcus aureus bacteria. What did they do in this study? They bring 4 patients here, 2 categories of patients and patients. الجروب الأول they give him 7.5 million Staphylococcus aureus, but they give it as injection with a solution, and they put it under 4 hours. And the second, so this is the first group. Second group, they bring the same amount of Staphylococcus aureus, 7.5 million Staphylococcus aureus, but they put it in gauze, piece of gauze, shashi, and they put it on the arm. So they wanted to discover, is it make a difference if we give a Staphylococcus aureus here on water for injection or normal saline, or if we give it in the gauze? The result was amazing, was really changed the way that the people think about how we will work on the group. For the first group that they take the 7.5 Staphylococcus aureus as injection, no one gets severe condition. Some of them they get mild symptoms, and all they already resolved. And there was in the group that they have it on this gauze on the mash. All volunteers, 100, experienced an infection, severe signs and symptoms. Some of them they need hospitalization, and the number of them they need a surgical intervention to remove the mash. What is the difference between these 2 groups? يا عمي ما احنا هون حطينا 7.5 million Staphylococcus epiderms, هون we already put 7.5 Staphylococcus epiderms. What is the difference in biofilm? لأنه in the second group, because we use the gauze itself or mash start to be the base for the biofilm. And this study redifferentiate our understanding for the biofilm. Here directly development of the biofilm make the biofilm get very tough characteristics when already dealing with the kinds of treatment. So here let's first here we talk about physical aggregation of the bacteria. There are physical connection happen between the bacteria, layers upon layers. طبقة فوق الثانية start to be developed. The bacteria immobilized. Now, in this case, if the bacteria can be mobilized or not, the biofilm no mobilized bacteria and aggregated. If we zoom in further, we can see that they are all attached and bound together to create cellular matrix of the biofilm. هذا community start to be developed here. And biofilm. When these layers, they start to build a ways of communication between each other. So there are an order that, oh, please, bacteria, produce more energy. Oh, this bacteria, please go up new bacteria come down and they start to make a special route for them. There are a connection of protein that will produce these layers, and the bacteria will move within these lines. Now, this biofilm characteristic element, this is really interesting topic in the wound care if you're looking for specialization, because there are variants in the wound care, and they start by the ways that the initial bacteria species developed in biofilm. The biofilm here, when it starts to be developed, usually you need a bacteria with a key, and this key will produce the first or initial call for biofilm. Then the bacteria from the wounds start to be connected to each other, and the way that the first bacteria here start to develop this biofilm can vary in the type of how the communication will work, how much the tunnels of roots, the kind of communication that will be varied. This is examples for another kind of biofilm. You can see the difference between two photos when it's already come from Staphylococcus epidermis and here from other ways that the connections, the way this is the roots, how it's already the bacteria will move on and communicate with each other. And this is not a dry environment. This is it's watery, it's full with H2O, so it's wet environment. Do you remember when we talk about destruction of the biofilms? Then the ones will be exudates. The exudates come from the destruction or removing this balance of water, because this is the communication, how it's already happened inside. So embedded in the matrix. And this creates the defense against the antimicrobial, against the white blood cells, the host layers, and so on. And also it will control the amount of oxygen, nutrients that happen, and so on. All this will be controlled. Now, really, it's very hard to tell how many layers can be in each biofilm. So we're talking about biofilm can happen in the chronic wounds with very small, 5 micrometer, but it can end by hundreds or thousands of layers of such bacteria. So you can see how much the resistance can happen on this kind of biofilm. So this is the challenging part that we have in the biofilm. How we will the characteristic of the wounds that will come with the biofilm, on mainly it's come with a shiny color. So you can see here the characteristic of the wounds that will be shiny in the color. Also, it will be easy to bleed. Usually, it's come with a mild exudate. And when the proper treatment starts and the destruction of the biofilm happens, usually they are expected that increase in the exudates that will come from these wounds. Usually, these wounds present a stagnated wound, so the wounds are not moved within whatever the medication or whatever the wound dressing that we have. Anyway, the biofilm takes also additional explanation and more. We will come to know more about the characteristic of the biofilm, inshallah, in the coming future. This is regarding to the bacteriology. Is there any question or answer؟ في أحد عنده سؤال أو استفسار؟ الله يعطيكم العافية يا ربي. If anyone has or gets a question about the biofilm, because this is really a challenging topic, it can take also, or may takes more time from us until we come to shape proper theoretical understanding how we will deal معاه أكثر. ممكن ياخذ معنا وقت أكثر لحد ما نقدر نعرف كيف بدنا نودي المريض the proper treatment pathways in dealing with such condition. الله يعطيكم العافية يا ربي. شكرا جزيلا لكم. سبحانك اللهم وبحمدك أشهد أن لا إله إلا أنت أستغفرك وأتوب إليك. هذا الدكتور عبد الناس فيكم استودعكم الله ونراكم إن شاء الله الأسبوع القادم. السلام عليكم.
Recording stopped.